Hello everyone, this is Depolina Das Gupta, a PhD student in Professor Shikha Laluria's lab, Department of Biochemistry, IISc. And today I am here to discuss with you all about an interesting topic, that is Saccharomyces cerevisiae, a model organism on which our entire lab works on, and also a powerful and popular system to study the fundamental aspects of eukaryotic cell biology. So right now on your screen you can see different images of Saccharomyces cerevisiae. The first one is how a cerevisiae looks under light microscope. The second one is Saccharomyces cerevisiae on agar plate. And the third one is a typical image of spot assay with Saccharomyces cerevisiae. So let's get started and get to know a cerevisiae a little better. Saccharomyces cerevisiae is commonly known as baker's yeast or brewer's yeast. In fact, the scientific term cerevisiae is also derived from an old world terminology for beer. It can ferment sugars to ethanol and carbon dioxide, an ability thoroughly appreciated by the brewing industry. The carbon dioxide released by the yeast has been used in the baking process for centuries to raise bread dough, giving it a light texture. Now coming to the scientific point of view towards a cerevisiae. It is classified as fungus or mold. A cerevisiae is a single-celled eukaryote. Yeast cells are approximately 5 micrometer in diameter, which is between a bacterial cell and a human cell. A cerevisiae also has another common name, that is budding yeast, and it derives this name from its characteristic feature of bud formation during cell division. This is the cartoon representation of a Saccharomyces cerevisiae cell. This is the mother cell and this is a newly formed daughter cell, which during the process of budding has left a ring-shaped deposit on the mother cell wall. These are chitinous deposits and referred to as bud scur. A similar kind of structure is also visible on the daughter cell. They are referred to as bud scur. Like any other eukaryotic cells, a cerevisiae also has different membrane-bound organelles like nucleus, cytoplasm, mitochondria, plasma membrane, and cell wall. In the previous lecture by Professor Shikha Laluria, she has explained to you all in great detail about the properties, structure, and function of all these membrane-bound organelles. Today, I will give you a brief idea about the cell wall. So what is cell wall? Cell wall is an extracellular matrix which in case of Saccharomyces cerevisiae is composed of a complex meshwork of beta-glucans, chitin and manoproteins. You can roughly divide the cell wall in two different layers. The inner layer is composed of beta-1,3-glucan, beta-1,6-glucan and chitin. Beta-1,3-glucan and beta-1,6-glucan are glucose polymers linked by beta-1,3 linkage and beta-1,6 linkage respectively. To give you a more detailed idea about these linkages, let's focus on their molecular structure. This is a glucose molecule with its numerical nomenclature. This carbon right here is the C1 carbon. The next one is C2, then the next one is C3 and so on. In beta-1,3-glucan, two consecutive glucose molecules participate in the glycosidic linkage by their C1 and C3 carbons respectively. If this glycosidic linkage is established between the C1 of the first glucose and C6 of the next glucose, then that is referred to as beta-1,6 linkage. Now coming to chitin. Chitins are biopolymer of N-acetyl glucosamine. This is the molecular structure of N-acetyl glucosamine, and in chitin, two consecutive N-acetyl glucosamines are linked to each other by beta-1,4 glycosidic linkage. The inner layer of the cell wall is responsible to provide strength to the entire structure. The outer layer of the cell wall is composed of manoproteins, which are nothing but glycoproteins. They determine the surface property of the cell. The cell wall as a whole is important to maintain the cellular integrity during growth, division, under stress condition, and also during the time of mating. The cell wall is also a primary target for different antifungal drugs. Another important function of cell wall is to maintain the cellular morphology. The S-cerevisiae cellular morphology or cell shape changes as the cell progresses through the various phases of the cell cycle. The first one being the establishment of polarization for bud formation during the late G1 phase. The activation of this mechanism is regulated by a complex machinery popularly known as the bud site selection machinery. The bud site selection machinery is composed of a number of integral membrane proteins which are inherited by the newborn daughter cells from their mother at specific locations on the plasma membrane. The bud site selection machinery then activates a downstream regulator protein called CDC42. The activated CDC42 then is recruited in a high concentration at the bud site, highlighted here in the yellow color. The CDC42 then employs a number of effector molecules which then regulate a number of downstream events. They are the recruitment of the septin ring, clustering of the actin patches, polarization of the actin cable and glucan synthesis. Together all these events leads to the formation of the bud. The green color here represents the septin ring. Septin ring is composed of septin proteins and is essential for the cytokinesis. They also regulate the positioning of the mitotic spindle during the cell division. The red dots here represent the actin patches. They are complex of actin filaments. Once the bud is formed, it keeps on growing in a polarized manner till the S phase of the cell cycle. This type of growth is often referred to as apical growth. During the G2 phase, a switch happens between the apical form of growth and the isotropic form of growth. The unpolarized form of growth is often referred to as isotropic growth. After the switch, though, the growth is still directed towards the bud, but now it is more evenly distributed inside the bud. And you can also see the regulator protein CDC42 for polarization is evenly distributed throughout the bud cortex. Now, after the apical isotropic switch, there is still asymmetry in the growth pattern of the bud and the mother cell. This asymmetry is broken at the late M phase, and the growth is now directed towards the bud neck, leading to the cytokinesis. The changes in the cellular morphology during the cell cycle is also regulated by different cell cycle specific proteins. For example, the establishment of polarization and bud formation is redundantly regulated by the G1 cyclin CDK complex. The apical isotropic switch is also controlled by the G2 specific cyclin CDK complex. Now let's move on and get to know some key features of Saccharomyces cerevisiae genome. In 1996, Geffu et al. First published the whole genome sequence of S. cerevisiae, which is a landmark for the entire eukaryotic biology. A typical haploid budding yeast cell genome is made of 16 chromosomes, containing approximately 12,000 kb genomic DNA. And you can actually visualize this chromosome in pulse field gel electrophoresis. In budding yeast, the genome has a very high density of protein coding genes compared to humans, which directly implies the concentration of intron is very less in S. cerevisiae. S. cerevisiae genome also harbors several dubious ORFs which are unlikely to encode proteins. The non-coding genes mainly encodes for tRNA or transfer RNA, rRNA or ribosomal RNA, and RNAs that are involved in several stages of gene regulation. The origin of replication in budding yeast are termed as ARS or autonomously replicating regions that are located at every 20 to 40 kb interval on each chromosome. The centromeres on each chromosome are known as point centromeres, because they are short, approximately 125 base pair in length, and they are not surrounded by heterochromatin like other eukaryotic centromeres. The telomeres in S. cerevisiae is composed of specialized DNA sequences and associated proteins. The telomeres, the rDNA locus that encodes for rRNA, and the silent mating cassets are the only regions that form a heterochromatin-like environment in the budding yeast genome. Now in this slide, I have introduced you all to a new term called silent mating cassettes. So what is this silent mating cassette? To understand that, let's first understand what are the different mating types in Saccharomyces cerevisiae. S. cerevisiae can exist in two different states, the haploid state and the diploid state. A haploid S. cerevisiae exists in two different mating types, MAT-A and MAT-alpha. A and alpha are pheromones secreted by the specific mating type of S. cerevisiae cells. Meaning a MAT-A cell will secrete A factors and MAT-alpha cell will secrete alpha factors. Each of these pheromones acts on the opposite mating type of cell and activate a signaling pathway often referred to as the pheromone signaling pathway. The pheromone signaling pathway in yeast is mainly composed of a 7 transmembrane G protein and MAP kinase cascade. Activation of the pathway is initiated by binding of the pheromone to the extracellular receptor on the cell surface. This in turn catalyzes the dissociation of G protein subunit G alpha from the rest of the protein components, that is G beta gamma. Now the G beta gamma is free and it can then in turn activate the downstream MAP kinase pathway. This leads to the transcription of specific genes followed by morphological changes, that is SMU formation, cell division arrest in G1 phase and ultimately fusion of two different cell types. This is the life cycle of the budding yeast. These two are haploid yeast cells of different mating types. The first one is the A cell and the second one is the alpha cell. Each of these haploid cells can divide mitotically by the bud formation and can yield more haploid cells of the same mating type. Under suitable circumstances, when these two haploid cells are in close proximity, they can undergo mating. During the mating, these cells releases the pheromone which causes the morphological changes in the cell, that is the SMU formation, ultimately leading to the mating process. The mating yields a diploid cell. This diploid cell can again divide mitotically and yield more diploid cells. Or under different circumstances, it can also undergo meiosis or sporulation, leading to the formation of 4 haploid spores, which upon germination forms 2 alpha cells and 2 A cells. Interesting factor is that these haploid cells can switch their mating type independently. Meaning a A cell can get converted into alpha cell and an alpha cell can get converted into A cell. So how does this switching occur? To understand that, let's focus on the genetics of the study. The mating type of the cell is determined by the MAT locus present on the right arm on chromosome 3. The MAT locus contains one of the two non-homologous alleles responsible for determining the mating type of the cell, that is MAT-A and MAT-alpha. In this case, it is MAT-A. Additionally, chromosome 3 has two more loci, HML-alpha and HMR-A. HML stands for homothallic mating left and HMR stands for homothallic mating right. These two loci contain additional copy of mating type allele. HML alpha contain MAT alpha allele and HMRA contain MAT A allele. These two loci are transcriptionally silenced because they are embedded in the heterochromatin region. During the mating type switching process, an endonuclease comes to the action. This enzyme is called HO endonuclease enzyme. They have their recognition site right here indicated by the arrow. The HO endonuclease introduces a double strand break at this site, which is then repaired by homologous recombination, where one of these two transcriptionally silenced loci is used as DNA donor sequence. After the repair by homologous recombination, the MAT-A allele is now replaced by MAT-alpha allele. The purple color box here is referred to as recombination enhancer. This is a DNA sequence which also regulates the homologous recombination. So initially we had an A type of cell with us and now it has switched its mating type to the alpha type. Interesting factor is that most of the laboratory budding yeast strains has their HO endonuclease encoded gene mutated. So most of them have their mating type switching restricted. Till now, we have discussed different cellular aspects of the Saccharomyces cerevisiae. Now let's try to understand why Saccharomyces cerevisiae is a popular and useful tool for geneticists. Saccharomyces cerevisiae has simple growth requirements. They grow rapidly in approximately 90 minutes under optimal laboratory environment. They can exist in both haploid and diploid states. They can be easily genetically manipulated, and a large database is already available for S. cerevisiae genome, referred to as S. cerevisiae genome database or SGD. There are also many popular molecular biology techniques which were initially standardized in Saccharomyces cerevisiae. The first one is the yeast-2 hybrid system. Here we uses a gene referred to as LAGZ. This gene encodes a particular protein called beta-galactosidase, which in the presence of XGAL can turn the yeast colonies into blue color. The transcription of this LAGZ gene is regulated by an transcription activator. This transcription activator has two separate domains. The first one is the transcription activation domain and the second one is the DNA binding domain. The transcription activator binds the specific DNA sequence upstream of the LAGZ gene by their DNA binding domain, and then the transcription activation domain activates the transcription of this gene, leading to the expression of beta-galactosidase. So this system is used to understand the interaction between two proteins. Let's consider two proteins. First one as protein X or bait protein and the second one as protein Y or prey protein. To understand whether these proteins are interacting with each other or not, we first fuse one of these proteins, for example protein X, with the DNA binding domain of the transcription activator, and then we fuse the other protein, that is the prey, with the transcription activation domain of the activator. Now, if these two proteins are interacting with each other, then the transcription activation domain will be in close proximity of the LAGZ gene, which will then activate the expression of the beta-galactosidase, ultimately turning the yeast colonies into blue color in presence of XGAL. If these two proteins are not interacting with each other, then the transcription activation domain will not come in close proximity of the LAGZ gene. Thereby, the transcription will not be initiated in LAGZ gene, ultimately leading to the white coloration of the yeast colonies. So just by observing the color of the yeast colonies, we can understand whether our two protein of interest are interacting with each other or not. So the next up is one-step PCR mediated tagging. Why tagging of a protein is important? If we can tag our protein of interest with a fluorescent tag, then we can visualize them under the microscope and get an idea about the subcellular localization of our protein of interest, which in turn will also give us an idea about the function of the protein. During the Western blot analysis, when the antibody against our protein of interest is not available, we can actually tag the protein with different epitopes and then use antibodies commercially available against these epitopes and visualize the protein on the Western blot. So let's understand how one-step PCR mediated tagging works. Here we have the plasmid with our tag of interest and the marker specific to the yeast cells. We amplify this entire sequence with a pair of oligo. The forward primer and the reverse primer have DNA sequences at their tail which will be homologous to the region where we want to incorporate this tag. We amplify this tag by polymerase chain reaction and then transform the PCR product in the yeast cell. The blue DNA sequence here is homologous to this region and the red DNA sequence here is homologous to this region. After homologous recombination, this entire cassette will be incorporated in the yeast genome and our protein of interest will be tagged. We can then screen for positive colonies by colony PCR and Western blot. In colony PCR, we design a set of oligo, one specific to the yeast genome and the other specific to the cassette. And during Western blot analysis, we can use antibodies against these tags. In the previous slide, I have mentioned that the plasmid contain marker that will be specific to the yeast cells. Now, in yeast cells, there are different categories of markers that can be used. First of all is the auxotropic markers. Genes that are necessary for the growth of the cell under an essential nutrient deprived conditions. Example, LEU2, HIS3, URA3, etc. Importantly, if we want to use these auxotropic markers, our strain should have a genetic background with the mutated version of these genes. The dominant markers are the genes that provide resistance to the cell against drugs to which the cell is sensitive, example norceothrysin, hygromycin B, etc. Selectable markers. These are the genes whose expression select for the positive transformants. And then comes the counter-selectable markers. These are the genes whose expression kill the positive transformants. Example, counter-selection for the URA3 gene involves 5-fluoroorotic acid. Cells that express URA3 gene accumulates a toxic metabolite like 5-fluorouracil in the presence of 5-fluoroorotic acid, rendering the cells inviable. Counter selection is important during plasmid shuffling. Plasmid shuffling can be used for various purposes, one of them being to understand the function of an essential gene. Generally, when we want to understand the function of a gene, we eliminate it from the genomic background. But in case of an essential gene, we cannot do that. So, we try to introduce different mutations in the essential gene and try to understand the function. Plasmid shuffling is the way we can do that. So, to begin with, we have a yeast centromere plasmid with us. This is also referred to as CEN plasmid. This plasmid contain a part of the sequence of ARS and centromere. Thus, they are autonomously replicating and single copy plasmids. This CEN plasmid contain a wild type version of the essential gene and URA3 marker. We transform this plasmid inside the cell. Now the cell has two copies of the essential gene, the genomic copy and the plasmid copy. Next we go for the targeted deletion of the genomic copy of this essential gene. Further, we take another CEN plasmid where we clone the mutated version of the essential gene. This plasmid has a different marker, that is HIS3. We again transform this plasmid inside the cell and plate these cells in 5-FOA containing agar plates. This will select for only those cells which will have the plasmid that contain HIS3 as marker. Thus, it also contains the mutated version of the essential gene. We can then rescue this plasmid and go for DNA sequencing to understand the mutation. This mutation can be a conditional mutation or a reducing function mutation. If this is a null mutation, then the cell will be dead. Next is tagging with green fluorescent protein or GFP. GFP was first isolated from a jellyfish called Achoria victoria. This is the crystallized structure of GFP and this is the protein data bank ID. Now, if you excite GFP with UV light, the GFP will emit bright green fluorescence. So, if we can tag our protein of interest with GFP, we can actually visualize our protein under fluorescence microscopy. So, to give you an idea how does it look under microscope, let's show you some images. These are Saccharomyces cerevisiae cells where we have tagged the NET1 protein with GFP. Now it is already known that NET1 is a nuclear protein that remains associated with the rDNA locus. These green dots here are the NET1 proteins tagged with GFP. Now there is an entire database called East GFP Fusion Localization Database that will help you to study the global analysis of protein localization in eukaryotes. This brings us to the end of today's short lecture. So far we have learned different cell morphology of Saccharomyces cerevisiae, their mating types and how do they switch their mating types. We have also learned some interesting factors about S. cerevisiae genome and why S. cerevisiae is so useful as a genetic tool. We have also understood different molecular biology techniques with Saccharomyces cerevisiae. Hope you all learned something new today and enjoyed today's lecture. So thank you and all the best.